Welcome to today's webinar. We will have two speakers today, Dr. Sarah Warren from NanoString, followed by Jason Frazier from Presage Biosciences. Dr. Warren will open by presenting a brief overview of NanoString's digital spatial profiling platform, GeoMx DSP, and the curated gene expression panels focused on biomarker and drug development. Dr. Warren is the Senior Director of Translational Science at NanoString Technologies, where she leverages multiplexed molecular profiling technologies to address key research areas in oncology, immunology, and beyond. Her role enables her to work with academics, biopharma, and clinicians to identify unmet needs in translational research and create novel products for transcriptional and proteomic profiling. She's also very active in the immuno-oncology research community, promoting the science and application of cancer immunotherapy to improve patient outcomes. Thank you so much for joining us today, Dr. Warren. We feel really lucky to have you. And with that, the floor is yours.
Thank you so much. I'm delighted to be here and to be able to tell you about some of the exciting new technologies that we're developing at NanoString. So as mentioned, I'm going to be talking today really about our drug discovery tools that we have for both the nCounter and the Geomix platforms. So in case anyone is not familiar with NanoString, we are a life science tools development company and research partner. We're founded and based in Seattle, Washington in the United States. And we have two platforms that we've commercialized to date. First is our nCounter system, which is used for bulk transcriptional profiling. And we've recently launched our Geomix digital spatial profiler as well for giving spatially resolved protein and transcriptional characterization of tissues. Both of these platforms are based upon our unique chemistry, which relies on direct hybridization of optical barcodes to the analyte of interest. So in nCounter space, we have applied the technology to be able to measure RNA, protein, and DNA. And in the DSP, digital spatial profiling space, we are able to profile RNA and protein. And I'll show you a little bit about how each one of those works. So first, for nCounter space, one of the really nice things about the platform is that it was designed to work on formalin-fixed paraffin-embedded tissues. And these are samples that are frequently available as part of translational or clinical research. And they're very challenging to characterize with other technologies that may be based more on enzyme-enabled amplification. So the nCounter system allows profiling of up to 800 distinct RNA targets. And we organize these into panels that are focused on a specific biology, or you have the ability to design custom panels for your own particular research question. But anyway, we have two elements to our detection technology, a capture probe and a reporter probe. Each of these are binding to RNA that has been purified from your tissue sample. And it also works with blood or cell culture or any other sample type where you are trying to characterize the RNA. And through this hybridization and then purification on our nCounter technology, we are able to count the presence of these barcodes within the sample, and each barcode corresponds to the presence of a single transcript of a single analyte. And so you can see that it's a quantitative digital counting mechanism to understand what that transcriptional profile is like in the sample that you're measuring. So we have applied this technology to all stages of the drug development spectrum. And you can see how we have designed a number of panels that we're listing on this slide to target specific elements along the research continuum, from discovery to preclinical development and clinical applications. So today we're really going to be focusing on that first stage, the new drugs and biomarker discovery. And I'm going to tell you about some of the nCounter panels that we have, as well as some of the Geomix reagents as well. So a lot of the biology that we focus on is related to oncology and immunology. And one of the panel series we have that's particularly useful in this space are what we call our 360 series panels. And these are very holistic panels that are looking at sort of total biology related to the tumor microenvironment, and the infiltrating immune system. But we also have panels that are focused specifically on different elements like particular signaling pathways, tumor progression, particular immune cell populations, or even metabolism. And in addition, any one of these panels can be customized with up to 55 spike-in genes that are user-selected to give a little bit more customization in terms of the research and data that are generated. We have developed as well a few clinical assays that can be deployed on the nCounter system in addition to our RUO assays. So these are the tumor inflammation signature, which is used for quantifying a suppressed adaptive immune response in the context of tumors. It is a way of selecting patients that are more likely to respond to anti-tumor checkpoint inhibitors. We have a PAM50 signature, which is for breast cancer subtyping, and we have a lymphoma subtyping signature as well. We've leveraged these clinical signatures into our RUO assays through some pre-designed reports that we have, the IO360 report or the BC360 report for breast cancer. So as I mentioned, we've are very excited about these 360 panels that we have. We've spent a lot of time curating the content, building out these associated reports with them, and really trying to embed within these panels as much interesting biology as the research community is going to find useful. We also have a third panel in this series called our tumor signaling panel that's very much focused on signaling pathways within the tumor. As I mentioned, we have, in addition to the research reagents, we also provide data analysis services. So we have the full 360 analysis reports, as I described, that will provide the scores for those clinical assays that we have from the research panels. In addition, it will characterize 27 different immune cell populations and a number of immune pathways in a very dynamic and interactive format. We can also calculate signal signature reports from our 3 clinical tests, and we also have the ability to do data analysis on any of our panels using our NSolver software that has been specially designed to facilitate these analyses. Each of these tools are coming from in-house experts that we have. It's generating publication-ready figures, and it's highly interactive for the end user. In addition to the 360 panels, we also have a wide variety of other gene expression panels, some of which I've listed on this slide. And you can see we're really trying to cover a broad survey of biology across different therapeutic areas, different research questions, even different species. We've called out specifically the canine IO panel here. Many of these other panels are also available for use with both mouse and human samples. And as I mentioned before, any of the panels that we have can be customized with up to 55 unique user-selected genes to really give that, that really focused research and data that's going to be specific for your particular research question. So here's just one example of how we use the nCounter panels for target discovery. And it's a particularly timely example because we're looking now in COVID-19 infected lung tissue. So last summer, when the pandemic was just emerging, we formed a collaboration with Dr. Robert Colvin at Massachusetts General Hospital. He had access to lung tissue from patients that had passed away from the COVID-19 infections, as well as patients who had passed away from influenza-infected lung tissue. He purified RNA from each of these, and then he characterized them with pan cancer progression and inflammation panels, and was looking for similarities and differences in the transcriptional patterns in both of those sample types as a way of identifying potential drug targets for therapeutic development. And I'm showing you just one figure from the paper that he had here, where he observed that there was a very unique T-cell population infiltration into the COVID-19 patients with a very distinct perivascular distribution within the alveoli. There were also unique genes associated with both the inflammatory response and vascular damage and repair, which manifested as angiogenesis pathways being activated. So this was a single experiment that he was able to do. He got a very nice publication out of it in New England Journal, and it was a very straightforward experiment and very clean data analysis, and you can see how easy it was for him to identify the really interesting biology that gave us more insight into the underlying mechanisms of infection. The other platform that we have for doing drug discovery biomarker development work is, of course, our Geomix and Geomix digital spatial profiling platform. This is, again, based upon that same barcoding chemistry, but we really wanted to be able to apply it now to understand where within different tissues we are seeing expression of particular targets. This is just a very high-level overview of how the assay works. We are, again, working with formalin-fixed paraffin-embedded tissues, although it's also compatible with fresh frozen or fixed frozen samples. And we incubate the tissue on a slide with either our barcoded antibodies or barcoded in situ hybridization RNA probes, along with fluorescent visualization markers. We then image the tissue on the Geomix system, select regions of interest for molecular characterization, and expose those regions of interest to UV light, which then cleaves the barcodes off of the analyte detection probes, captures them in a microtiter plate, where they are then quantitated either via nCounter or via NGS sequencing. So the real heart of the technology is based on our ability to control the path of that UV light. We do it using what we call a digital micromirror device that is a million little tiny mirrors that are each independently controlled and can be programmed by the computer to either turn on, which will let the light pass through the tissue, or be off, which doesn't allow the UV light to go through. And what we can do then is use the fluorescent images that we're generating to visualize the tissue to make automatic masks that will control that path of UV light. So in the top slide, in the top right side of the slide, you can see the tumor-specific mask that we've made by passing the UV light only through the tumor cells of this particular sample, which are the yellow cells that you can see in the middle image. And then we collected all of the barcodes from that mask, we put them in one well of microtiter plate, and then we went back to that same tissue, that same region of interest, and we inverted the mask to make a tumor microenvironment mask. And now we're collecting all of the barcodes from the pan-CK negative cells that are not the tumor that are present here. And we collect those into a separate well of the microtiter plate and quantitate them independently so we can really understand where within this tissue we're seeing particular targets being expressed. And here's just what that data can look like when you do that. On the right side of this graph, we were using the Geomix to characterize this particular sample with a 96-plex RNA assay, and you can see very clearly, without even being able to read the gene names, there's very distinct molecular patterns associated with the tumor cells and the non-tumor stromal cells within this sample. So one of the things that's been really powerful about this technology is that we've been very creative about how we deploy the ability to draw these masks on the tissue. And so I'm showing you here 5 different visualization strategies. The first and simplest is our geometric profiling, which is just where we collect everything that's within that region of interest as a single sample point, and then can compare it to other regions of interest within the same tissue or across different tissues. Second is our segmentation approach, which I previously described to you. And then our rare cell profiling approach is just a variation of segmentation. But here, rather than defining whole tissue compartments, we are using fluorescent markers to illuminate particular immune cell populations, such as CD3 T cells or CD68 positive macrophages. And we collect the transcriptional or proteome profiles from each of those cell populations independently. Contouring is just as it sounds, where we are defining regions of interest based upon their distance from a user-selected point in the tissue sample. So we might use it to characterize distribution gradients across the tumor microenvironment, or distance from an apical surface within a stratified epithelial layer. Graded approach then is the last profiling mechanism we have, where we are doing unbiased sampling of a region of interest to collect the entire transcriptional profile in distinct individual wells so that we can more finely dissect exactly where those signals are coming from. None of this profiling would be possible unless we had the appropriate panels for looking at the analytes that are of interest to our users, and so we have developed a number of protein panels for the platform. The two that have been most developed to date are one for cancer and immunology, and a second for neuroscience. And each of these are organized into a core plus module format, where you order the core target set, which also contains all of the controls that you will need to run the assay. And then you can customize it with additional content specific for your research question by adding one or more modules. All of the antibodies that we are including in our Geomix platform have gone through an extensive testing validation process, so you can have a lot of confidence when you start your Geomix experiment that those antibodies are going to work right off the bat for your experiment and not need a lot of development work in order to generate good data. The RNA space is another aspect of the Geomix platform that we have been developing extensively recently. We launched our Cancer Transcriptome Atlas in the middle of last year. This is a panel that contains 1,800 different genes for characterizing tumor biology, again, that immune response in a microenvironment. So in many ways, it's very similar to the 360 panels that I talked about for nCounter, but of course it contains a few more genes, and you have the ability to customize those targets. And as soon as we launched this panel, we realized that there were investigators who had questions beyond cancer and immunology, and they really needed other solutions. So we went back and we developed our whole transcriptome atlas now, which was for characterizing the whole transcriptome, basically 18,000 genes in humans, 22,000 genes in mice. It's a non-3' biased whole transcriptome profiling because we're relying on that direct hybridization of the probes to the target. So with this, we can get complete coverage of the transcriptome. And again, we have the ability to do customization here, which allows us to characterize non-endogenous genes, exogenous genes, synthetic targets, non-coding RNAs, whatever is of interest for you. And so one particular application of that high-plex RNA profiling panel is again in the COVID-19 space, where we were working on a collaboration with Dr. David Ting, again from Mass General, as Bob Colvin was involved in this project too. And here we were characterizing lung autopsy tissue from COVID-19 patients that either had a high level of virus detectable at the time of their death or low levels of virus. And we did characterization on these samples with both the CTA panel and a 60-plex protein profiling assay. I'm just showing you one data figure from the paper where they were using RNA scope from ACD to visualize the actual virus present in these samples. And that's what you see on the far left side of the slide, the white and pink image. And then we selected in genomics space regions of interest that were virus high or virus low from that tissue. And then we characterized the transcriptional differences between these two different ROIs across 5 different patients. And what you can see there on the right side of the slide is the volcano plot that shows the differential gene expression that was associated with presence of detectable virus. In short, we saw a lot of chemokines, interferon signaling associated with the virus, but there was not the expected localization of T and T cells, B cells, and K cells in those same regions of interest, which suggests that there's a dysregulation in terms of the immune cell trafficking going on in these samples. These results were published recently in Nature Communications just a couple months ago, and we look forward to being able to share more stories about how Geomix is being used for COVID-19 profiling in the very near future. If you're interested in that story or any of the other stories showing Geomix applications, I'd encourage you to go to our website where we have links to the 38 publications today that use GeoMx technology, and you can see just a selection of those journals that are publishing Geomix papers here, and you will very quickly notice that they're very high-tier journals across a wide variety of fields. So I encourage you to go and find the stories that match closely with the research that you are interested in doing. And with that, I'm going to wrap up my portion of the presentation, and I leave you with our vision for how NanoString platforms can be used in the drug discovery process. We really think that there's a place for both profiling and spatial profiling to form a research continuum where we are looking back and forth across both axes to understand how potential drug targets, how biomarkers, and how genes and proteins that are controlling the fundamental biology are distributed within the clinically relevant samples that our users are able to access and interested in profiling. So thank you so much for listening to my talk today, and I'm very pleased to be able to share the work with you.
All right, we'll now turn to our next speaker, Jason Frazier of Presage Biosciences. Jason is the Director of Scientific Operations at Presage, a Phase 0 clinical research organization. He oversees Presage's testing lab and preclinical research lab operations in support of partner-sponsored clinical and preclinical research. He has spent the past 10 years at Presage working to develop and expand their assay technologies. Prior to joining Presage, he worked as a scientist at Targeted Growth and at Rosetta Informatics, Merck Research Laboratories, working on development of a lentiviral high-throughput screening platform. He will be speaking today on deep mechanistic profiling of immuno-oncology drug combinations in cancer patients using two complementary platforms. Thank you so much for joining us today, Jason, and the floor is yours.
Great, thank you. Hello, everyone. Thank you for joining the presentation today. So first, I'd just like to take a minute to acknowledge and say that I think we can all agree that we're very excited about technologies like the digital spatial profiling capabilities in the Geomix system that is being developed at NanoString. They're incredibly powerful in that they enable greater understanding of complex molecular interactions within the architecture of tissues, including cancer. And so today, it's my pleasure to speak to you all about how we are using this high-plex biological readout technology and using it to profile samples and applying this in our early phase 0 clinical drug development studies. The data I'll show you today started really with a question, and that is, what if you could combine spatial profiling with a way to precisely expose distinct areas of a tumor to different drugs and then evaluate those responses? And really thinking about this, if this is feasible, thinking about the impact that we could make on translational oncology and early drug development, and think about what such an approach could mean for even the future of precision oncology. And I know that there are certainly others out there who have thought about this. So in 2016, the Cancer Moonshot Report was published by the NCI and leading experts to put forth recommendations on how to accelerate cancer research and discovery efforts. And in this, they identified some of the unmet needs in key areas such as translational research, where currently the rate of success in translating drug development programs guided using preclinical models to approved therapies is still extremely low at around 10%. In this, they describe the potential solutions to this problem, including developing dependable platforms on which to test anti-cancer agents to accurately predict success in patients, including devices that can deliver microdoses directly into intact patient tumors. This being a technology that would allow us to better understand the response of the intact microenvironment and immune system during research in translational drug studies. And at Presage, we have developed such a platform, and the platform that we have developed we call CIVO. So CIVO stands for Comparative In Vivo Oncology, and as its name implies, it's a technology that allows drug developers to investigate and compare responses to drugs or combinations of drugs within a living tumor while the tumor is still intact and in the patient. And as you can see in the upper left-hand graphic, it's a multi-needle injection device that is capable of accessing living tumors and delivering up to 8 different microdoses of investigational drugs or drug combinations directly into the patient's tumor. The injection results in distinct and spatially defined columns of drug within the tumor, as you can see illustrated in the middle left panel. And an important aspect of to the technology is the co-delivery of fluorescent microspheres, which we call CIVO glow, that remain localized at each site of the injection and help us to uniquely identify each injection site for downstream sample processing and analyses. Once the tumor is surgically biopsied or resected, the sample is then cross-sectioned perpendicular to the injection plane, as you can see in the lower left panel. And we can then use these samples to generate thin sections for more traditional immunohistochemistry to evaluate biomarker responses, like you see in the central larger image. In blue, you can see the nuclear DAPI stain, and in green, you can see the more punctate fluorescent microspheres that denote each injection site, which helps us identify each of these sites, even in cases where we cannot see an apparent response to the drug. And in red, you can now see one of our tried and true biomarkers of response, cleave caspase-3, which shows us which of the drugs may induce tumor cell killing through pathways such as apoptosis. We have a number of different IHC and in situ hybridization stains that we're able to leverage to assess responses, including markers of cell death, signal transduction, immune cells, and also activation status of immune cells. So now looking at this slide here, you'll see in the right-hand side an image of an example of a CIVO microinjection site in a patient tumor where the drug has induced a classical marker of T-cell activation, granzyme B. And upon closer look, we even find cytotoxic T-cells throughout this injection site and drug exposure zone attacking the cancer cells. So now with the CIVO technology, we are addressing some of the key components for improving the success and efficiency in translational oncology, including these sorts of comparative analysis of drugs in these spatially defined ways and within the native tumor microenvironment and intact immune system in patients. And we're now able to do this in patients with investigational new drugs and drug combinations with investigational new drugs. Given the spatial aspect of an approach like ours with CIVO, I can see, I think it's easy to see why our technology is nicely complemented by a technology like NanoString's digital spatial profiling. The ability to now spatially profile samples like the ones that we generate exponentially increases the number of biomarker reads we can get from each one of our precious clinical samples. So now I would like to talk about how we have begun using this high-plex spatial readout to cast a highly targeted but much broader net to evaluate tumor response in CIVO-injected patient tumors. By combining these two spatially resolved technologies like CIVO and Geomix, we can now look at how individual patient tumors and immune cells respond to different drugs in this sort of high-plex fashion. The ability to retain the architecture of the tumor in our samples is absolutely necessary for the type of spatially defined analyses we do with our CIVO approach. And this is exactly why we like digital spatial profiling and why we've begun layering geomics analysis on top of our CIVO multiplex drug analysis using the sort of high-plex profiling panels such as the Cancer Transcriptome Atlas that Sarah mentioned earlier, gives us far greater information from each sample than the more traditional low-throughput immunohistochemistry and in situ assays that one can run on these types of samples. So now by combining CIVO and Geomix, really if you think about this, it's as if we're putting a single patient into 8 different arms of a clinical study at once, and then have the capability to profile how their tumor is responding to each of the different treatments, and now even how their immune system is responding as well. So to take this sort of synergistic platform technology a step further, the Geomix platform really allows us to stain for rare cell populations, such as T cells and other immune cells, and apply this digital spatial profiling specifically within these populations to compare how they are responding to the different drugs across the different treatments. So using the DSP panel, like the CTA panel, which consists of over 1,800 gene targets, we can now generate upwards of over 30,000 different potential biomarker readouts in a single study. And we can do this using only a single tissue section, which is really of value to us since it allows us to preserve a lot of our precious clinical samples. So as we've begun to study drugs that have immunomodulatory properties in our clinical studies, we have found that using earlier time points and transcriptional readouts gives us far more information on the activation status of the immune component and the tumor microenvironment than we typically would see running standard immunohistochemistry and in situ assays, which are far lower throughput and low plex. So now that I've shown you how we are combining these two technologies, such as ours with CIVO and the NanoString DSP profiling, to investigate the tumor microenvironment and rare immune cell populations, I can show you how we've used this approach to evaluate responses to some of these more interesting immunomodulatory drugs like checkpoint inhibitors. So this is particularly exciting application of these two technologies, as we can now begin to gain more mechanistic insight into how these types of drugs may act and act together in combinations in patient tumors without having to expose patients to the typical systemic administration and levels of these types of powerful immune modulating drugs. So in the clinical setting in soft tissue sarcoma, we've begun evaluating checkpoint inhibitors, including drugs like nivolumab, an anti-PD-1 antibody drug. And we do, in fact, see responses that are specific to the mechanism of action of a drug like nivo, including the activation and cytokine secretion of immune cells, specifically within the anti-PD-1 drug injection sites. As you can see here on the right-hand panel, we see some specific responses in the anti-PD-1 versus a more traditional classical chemotherapy in soft tissue sarcoma, trabectedin. Upon relieving T cells from checkpoint, we can see their activation and secretion of cytokines such as interferon gamma. And when we stain our sample to look further downstream at activation of macrophage and dendritic cell population, we can see things such as an increased production of related chemokines like CXCL9 and CXCL10. So traditional immunohistochemistry and in situ staining provides us with some insight into the activation status of the immune component of the tumor microenvironment, but it is definitely not the whole picture. And so we've been looking for ways to evaluate biomarkers of checkpoint inhibitors across cell types within the tumor microenvironment, and looking for ways to evaluate many potential markers of activation within that population. So naturally, this leads us to asking, can we focus our analysis of CIVO samples on specific cell populations and figure out what other gene transcripts are changing in these populations as they are exposed to different drug treatments, such as specifically the T cell component. The rare cell profiling capability of the Geomix system allows us now to directly answer this question. And so using, again, a CTA panel, we can do in-depth profiling of not only the T cell compartment, but also the adjacent tumor cells, and really any cells that are present within the tumor microenvironment. From this, we are now generating numerous biomarker readouts that will help us inform how these cells are responding and activating when exposed to drugs like checkpoint inhibitors. This is a look at some of the data from the first time we applied rare cell profiling to one of our soft tissue sarcoma clinical samples. And we were happy to see that we did in fact confirm what we had expected to see. Using a CD3 masking strategy in Geomix, we showed that in our samples, we could enrich the gene expression readout for not only the CD3 gene transcript, but also for many T cell and inflammation associated genes. So in the left-hand violin plots here, you see that the CD3 masking strategy works, and it works quite well. And we can show that enrichment of the CD3 transcript within the CD3 specific mask and not in the inverse tumor mask. And now taking this another step further and looking deeper into the data, in the right-hand volcano plot, you can see the CD3 mask samples are again highly enriched for T cell specific transcripts like granzyme B and CD8. And in the tumor cell mask, we see enrichment for transcripts that are typically more associated with tumor epithelium and many of the other cell types that are in the tumor microenvironment. So what happens when we apply rare cell profiling and now look more broadly across multiple different drugs and drug combinations in the same patient tumor? So you can see here in this slide, this is a breakdown now of the top 100 or so genes that are regulated across the different treatments within the different compartments of this patient's tumor. So you can see here in the green rows, the CD3 positive cell masking. In the inverse CD3 negative cells, or any cell that is not CD3 positive, you'll see that in the yellow rows. And the combined CD3 and CD3 negative cells, that would be basically all cells within the tumor and tumor microenvironment. You can see that as a magenta row in the figure. And in the left-hand section of this particular heat map, you will see the top mostly down-regulated genes across treatments. That's shown mostly in the blue colors. In the middle section, you see the genes that are more variably regulated by treatment or cell type. And then on the right-hand section, you'll see the genes that are strongly upregulated by most treatments, as you can see here in the darker red colorations. Upon closer look at this data now, we see some genes that are highly regulated specifically by treatment and also by cell type. So now taking a much closer look again at this data, in the green circle, we see significant upregulation of granzyme B in the T cell compartment of the IO combination treatment site, which included aldesleukin and nivolumab. And unexpectedly, in the yellow circle, we saw significant upregulation of IDO1 in all compartments in this particular combination of aldesleukin and nivo treatment. This is potentially a significant finding in this patient's tumor, given IDO1's role in tumor immune tolerance and the potential as an early mechanism for resistance. So again, looking at this a little more specifically and drilling down even a little deeper into the data, again, in the plot on the left, we can highlight and see the T cell specific elevation of granzyme B at the site of aldesleukin and nivolumab combination. And we see this across multiple levels of the patient tumor that we analyzed. Similarly, in the right-hand plot, we see increased expression of IDO1 within the tumor microenvironment across now multiple tumor levels, And we see this at sites of aldesleukin, at sites of nivolumab, and with the most significant elevation of IDO1 occurring within the IO combination site itself. So based on these findings with the Geomix digital spatial profiling data set that we were able to produce, we then took this and validated some of these observations using some adjacent tissue sections and orthogonal assays such as in situ hybridization for either CD3 and granzyme B or IDO1 itself. In this slide here, I just point out, you can see the increase in the granzyme B expressing T cells specifically at the site of the combination of aldesleukin and nivolumab injection in the CIVO injected tumor. And similarly, we were also happy to see that we confirmed upregulation of IDO1 via in situ hybridization staining and quantitated this particular response using image analysis. The overall IDO1 upregulation in this patient tumor at the site of the IO combination is significant compared to all of the other treatments in this patient's tumor, including another injection site that contained a combination of doxorubicin, a more classical chemotherapy, and nivolumab. And this comparison is seen when you are now comparing the two green bars in this bar chart. So confirming the spatial profiling gene expression results through these orthogonal assays gives us a lot of confidence in this type of approach and this type of high-plex analysis. And so we were very pleased to see that we were able to confirm the results through our labs and through these assays. So now, just to wrap up here briefly, we're excited about these two technologies and the way that we can combine them for translational research. We're really using them to begin to understand and gain insight into some of the early mechanism of action of drugs, like checkpoint inhibitors, as well as the tumor microenvironment response to these types of drugs when we introduce them directly into unique patient tumors. And using the data like I just showed you, we can begin to uncover potential actionable hypotheses for how to combine drugs, not only for drug development purposes, but really in the future for precision oncology purposes. So I'll end there. Thank you for joining the presentation today, and thank you to everyone at NanoString and the NanoString Tech Access program for their support in helping us generate genomics data here early on. And of course, a big thank you from the Presage team to all of you for joining the talk today.
Oh, excellent talk. Thank you so much, Jason. Really appreciate it. If you don't mind, I have a few questions for you. First off, what is the typical time point you evaluate tumor response for the CIVO microdose drugs?
Yeah, that's a good question, and that's an important question as it relates to the types of drugs that we're studying. So typically in our clinical studies, we are able to remove or biopsy tumors within an hour and as far out as 120 hours following the intratumoral CIVO microdosing. And largely this time point depends on the mechanism of action of the drug and the type of response that we're looking to evaluate. So for example, we would select maybe a shorter time point for the evaluation if we're interested in looking at early biomarkers of resident immune cell activation.
Great. Another question here. Can you explain what you mean by phase 0 clinical studies?
Yeah, this is probably a less familiar phase in clinical and translational studies. Phase zero studies are clinical trials that have been developed in response to the exploratory IND guidance put forth by the FDA. And this guidance allows researchers to administer subtherapeutic microdose quantities of investigational new drugs. And so we've been able to leverage this guidance using our CIVO platform in a unique way, in that we can deliver this microdose directly to the tumor and achieve these spatially resolved higher concentrations of the drug without systemically exposing the patient to that drug.
Fantastic, great overview. And one final question. How large are the spatially restricted drug response zones in the tumor tissue, and how do you select your areas for analysis, such as placement of AOIs or areas of interest for DSP?
Yeah, so the typical drug response zone or drug exposure zone within that column of drug that we introduce into patient tumors is about anywhere from 3, sometimes 4 millimeters in diameter. The co-injection of our CIVO glow microspheres, which I pointed out earlier in the talk, they're these small fluorescing microspheres that stay localized at the sites of injection. These really enable us to identify each one of those drug exposure zones or injection sites after we cross-section the tumor and after we thin-section the fixed tumor for immunohistochemistry and other assays like Geomix. So by using these fluorescent tracking microspheres, it allows us to find each injection site and then place areas of interest like we do in the Geomix system. And in some cases, we can leverage an adjacent tissue section that maybe has a stain of particular interest for a biomarker that we can also leverage to place areas of interest for types of analyses like Geomix.
All right, great. Thank you so much. And with that, I think we're at time for the presentations, but I wish to thank both speakers again so much for sharing their insights with us, and I hope everyone has a good day.